Biotechnology, in the broadest sense, means changing living structures to produce something new. In a real sense, the most powerful biotechnology first arrived on planet Earth about 4 billion years ago. Today, the products of this first biotechnology can be seen everywhere, in the air, in the earth, and in the water. The story of how that technology has worked out over these 4 billion years is becoming well known on Earth. It is called evolution by natural selection. As the third millennium begins, scientists have learned some of the secrets of how nature manages this bioengineering. And today we are beginning to do some bioengineering of our own that is changing the Earth in surprising and potentially revolutionary ways. By far, the most important success of biotechnology on Earth is the existence of life itself in all its surprising variety. Earth's first biotechnology arose in the ancient ocean, where atoms like carbon and oxygen and hydrogen, sulfur and phosphorus and nitrogen bonded together to form molecules that stayed together long enough to replicate themselves over and over and over. At present, we are puzzled as to the details of just how that happened, but we are confident it did happen in ways we are now beginning to understand and even able to replicate. Now, one of the important things to notice is that nature's engineering, producing a cell, a tissue, and an organism that can stay alive and reproduce, is bioengineering at the atomic and molecular level, an atom, a molecule at a time. Scientists have discovered one of the most important keys to this atomic molecular engineering, and that key is the structure of a large molecule called deoxyribonucleic acid, DNA for short. DNA, biologists discovered a few decades ago, is shaped like a twisted ladder, a doubled helix. The rails of the ladder are sugar and phosphate groups, and the rungs are pairs of 4 common bases: guanine, adenine, cytosine, and thymine. And it is the sequence of these base pair rungs that are the codes to direct the assembly of a living thing. Any living thing and all living things, atom by atom, molecule by molecule. This code is how nature puts together bacteria, cells, tissues, wings, legs, hearts, eyes, brains, amoebas, oak trees, snakes, beetles, butterflies, giraffes, whales, chimpanzees, and human beings. Well, when human beings first evolved on Earth some 3 million years ago, another new twist was added to bioengineering capabilities: the birth of intelligence, conscious thought, rational control. Using this new capability, humans managed to develop the first consciously programmed bioengineering feats. They discovered how to do things like select and modify seeds of wild plants for deliberate culturing in gardens and fields. And they learned how to tame wild animals and use their products like milk, meat, protection, and energy for human purposes. And the first agricultural societies were born. Now, this was bioengineering at the whole organism level. Humans got closer to the cellular level, though as yet they knew nothing of cells, when they learned how to ferment grains and fruits to make beer and wine, how to culture milk to make cheese and yogurt, and how to dry and salt meat for long storage. Over the centuries, they improved on all these early biotechnologies. They began to deliberately cultivate and breed new kinds of plants and new kinds of animals, living things never before known in nature. And they learned how some kinds of animals and plants were harmful to humans, and they invented ways to destroy them. Parasites like the pneumonia bacteria, the tuberculosis bacteria, poliovirus, smallpox virus, and many others. All of these early biotechnologies worked on a level of cells, tissues, and whole organisms, but with little detailed knowledge of how the technologies actually worked. Well, in the early 21st century, Such detailed knowledge is rapidly becoming available, and work on these levels is continuing and producing some dramatic breakthroughs. Cloning, for instance. Ordinarily, living organisms on Earth reproduce by sexual methods. The egg from a female combines with the sperm from a male, and the resulting fertilized egg develops into an embryo and then into an adult of the same species. The hereditary codes are carried in the egg and sperm in packets called genes. These genes are themselves sequences of base pairs on the DNA molecule. In normal reproduction, both the egg and sperm each contain only half the adult number of genes, but when the half number from the sperm combines with the half number from the egg, the resulting fertilized egg and the resulting baby and adult is thus a completely new and novel mixture of genes. Well, for centuries, humans have known how to clone plants by taking non-sexual cells and tissues from the roots or leaves or stems, culture them in soil or water, and grow new plants that are genetic duplicates of the parent plant. Clones, that is. Well, multicellular animals were harder to clone. Since almost all cells in an adult animal have the same complement of genes, just like plants, biologists long ago speculated that it might be possible to reproduce an animal from a cell other than the sperm or egg. Why couldn't you, for instance, take a skin cell from a mouse and use the information coded in its nucleus to produce a new mouse that would be exactly like the original mouse, or a cow, or a sheep, or a human? Well, in 1994, biologists in Scotland succeeded in cloning the first mammal, a sheep. Using special microscopes and micropipettes and probes, they took the nuclear material, which contained the DNA codes, from a mammary gland cell of an adult sheep. They carefully inserted this nuclear material into an egg cell of another sheep, from which the original nucleus had been removed. Well, after many failures, they found success, and the first cloned mammal, Dolly, was born. Since that time, over 500 other mammals, sheep, cows, goats, mice, have been cloned successfully. As yet, no human being has been cloned artificially, but that possibility is there, and it may be realized in the 21st century, even though many human governments around the world have already passed laws prohibiting such biotechnology. Getting closer to the atomic molecular level of bioengineering, humans are now experimenting with changing the genetic codes of plants and animals and bacteria. Nature has been doing this for 4 billion years. The results from nature's experiments are the present-day array of over 3 million separate species of plants and animals that inhabit the air, earth, and water, as well as the 10 to 20 millions of species that once lived on planet Earth but are now extinct. In laboratories like this one, human scientists are using machines like this newly invented gene gun to forcibly insert new genes into plant and animal embryos. When successful, the emerging organism will be different from its parents in particular ways that humans find helpful. For instance, genetic engineers have successfully inserted a special gene into soybeans that makes the plant resistant to specific herbicides. In the past, soybean farmers could not use the most effective herbicides to kill weeds in their field because the same herbicide that killed the weeds would harm the soybeans. Instead, the farmer would need to work the field 6-7 times a summer to keep the weeds at bay. Now, with a new genetically altered soybean, they can use the more effective herbicide only once or twice a season, greatly reducing their workload, saving fossil fuels, producing more soybeans per acre, delivering a superior product at the end of the season, and potentially saving more of the Earth's surface from agricultural encroachment. In other words, helping to preserve more wilderness areas and save more species from extinction. Over half the soybeans grown in the United States now use these genetically altered soybean seeds. There's a similar story with corn. a genetically engineered variety that needs less fertilizer and less herbicide to produce more corn per acre is now in wide use around the world. And one of the most important of the world's food grains, rice, is being developed with a special gene inserted to make vitamin A in the edible grain. This variety may turn out to be very important for consumers in some of the poorer countries on Earth, where blindness from lack of vitamin A is a serious problem. Work is also proceeding to add a gene to rice that would make it richer in iron. This would make the rice able to combat anemia, another widespread problem among women in poorer countries of the world. Just one more example. One of the world's greatest killers today is hepatitis B. It can be prevented, though, with a 3-shot course of inoculations that cost about $200. Well, most of the deaths from hepatitis B occur in poor countries where this cost is out of the question for most people. Well, researchers in the United States are having success now with inserting the genes that code for hepatitis B vaccine into a banana plant. If and when commercially successful, a life-saving inoculation for this disease, eating a banana, would cost less than 10 cents. In addition to genetically altering crop plants for more production, more nutrition, and for disease control, scientists are also inserting genes into plant species that can make them powerful pollution controllers. Plants are being genetically engineered, for instance, to make them able to soak up cadmium, copper, mercury, or arsenic compounds from the soil and bind them into their tissues. Then you harvest the plants and bury them where they can do no harm. Genetically engineered plants are also being developed that can produce industrial chemicals for a fraction of the usual factory cost and with much fewer and less difficult pollution problems. Biotechnologists around the world are combining gene engineering with cloning techniques. Cows, sheep, pigs, and mice, for instance, have all been genetically altered to produce particular hormones, enzymes, proteins, antibiotics, or other useful chemicals. These genetically altered animals, called transgenic animals now, are then cloned. Potentially, you have a whole herd of transgenic cows, for instance, that produce large quantities of human insulin in their milk. Or a whole farmyard of transgenic pigs that can provide whole organs able to be transplanted into humans. Or on a different level, the cellular level, a rich harvest of bioengineered human blood cells able to fight and defeat the AIDS virus. Or maybe even able to slow or stop the chemicals that cause Parkinson's or Alzheimer's disease. Some of the most important and most promising experiments in biotechnology on Earth use yeast cells or bacteria for their subject. Most people are not aware of the enormous populations of bacteria and the enormous number of different species of bacteria that far exceed the populations and species of multicellular plants and animals. In the human mouth alone, there are over 600 species of bacteria have been found. Well, some bacteria cause disease in plants and animals, but most bacteria perform essential jobs in cycling elements in the ecosystem of planet Earth. And bacteria are ideal subjects for the biotechnology lab. They're easy to engineer, easy to culture, fast to reproduce, and versatile in producing useful products. In biotechnology laboratories, scientists have learned to cut and paste genes into the DNA of bacteria, creating designer bacteria that can carry out specific tasks useful to humans. A common procedure is to insert a gene into the bacterial DNA, called a plasmid, and then shoot this plasmid with a gene gun into a plant or animal embryo. Scientists are also actively designing experimental, custom-to-order bacteria. Bacteria that can make insulin. Bacteria that can make spider silk. Bacteria that can clean up oil spills. Bacteria that can make human drugs. Bacteria that can make an unlimited number of enzymes, proteins, and other chemicals useful to human well-being. Well, this fast-growing parade of designer plants, animals, yeast, and bacteria is not coming to Earth without controversy. Critics are alarmed at the wide and sudden acceptance of what they consider a radically new technology. Many argue that there should be more testing. How will the sudden introduction of these new designer bacteria and yeasts and new designer plants and animals affect the Earth's ecosystems? How will the food produced by genetically altered corn, soybeans, rice, and barley affect human health? As yet, claim some critics, no long-term studies have been done by the corporations that are producing these popular with farmer crops and these popular with corporation chemicals. They may save the farmer some work and result in less herbicides and pesticides and fossil fuel use even, but what will they do to the end consumer? They may be useful to the chemical industry, but what about the environment?
In Europe especially, these critics have had a strong influence on government policy and popular acceptance. Most experts, however, think that the short and long-term benefits of biotechnology to producers, to consumers, and to the environment are so obvious, and the hard evidence supporting those benefits so strong that the 21st century on Earth will be the century of biotechnology. The most ambitious of the new genetic research projects is called the Human Genome Project. The first phase of this large-scale international study is now finished, and humans have rough drafts of the entire human genome. That is, they now have computer records showing the exact sequence of all the base pairs on all the human chromosomal DNA. The next step will be to identify all 20 to 30,000 human genes on the DNA, and to figure out just how these genes do their directorial tasks. Well, besides the Human Genome Project, in many sister projects, scientists have decoded the genome of many other organisms, from yeast plants to fruit flies, from corn plants to mice to chimpanzees. One current project is even trying to decode the genome for one of our human ancestors, the Neanderthal man. Comparing our genes to that of our close relatives, like the chimpanzee and the Neanderthal, will lead to powerful new insights into the evolution of life on Earth.
In these new research projects, we are rapidly approaching the atomic molecular level of bioengineering that nature has used for 4 billion years to produce the world's biosphere as we know it today. And when we learn not just the basic code, but the details of how that code operates inside living cells to produce the proteins and enzymes that then produce the cells, tissues, and organs of living creatures, there is little limit to what the future may hold. At this atomic molecular level, the sciences of physics, chemistry, biology, and ecology are all merging to form a basic level science of life in the universe. Physicists and electronic engineers, for instance, are approaching this level now with what they call nanotechnology. Nano means very small, technology at the atom and molecular size level. This scientist at IBM, for instance, has already shown how it is possible to control a single atom, moving that atom at the will of a human being to form structures one atom at a time. This scientist at the University of Arizona has found a way to build a new molecule of carbon called Buckminster fullerene, buckyballs for short. And other scientists have been able to make nanotubes of carbon, the most important of the basic life atoms. Well, just as the molecule DNA can direct the activities of a cell, including the reproduction of that cell, so scientists are now trying to design molecules, often with a life-centered carbon at their core, that can count and reproduce themselves, making for the possibility of nanocomputers. These computers would be microscopic in size able, for instance, to flow through the bloodstream of a person and do special jobs inside. Jobs like finding and destroying malignant cancer cells, or jobs like finding and fixing defective kidneys or hearts or lungs. Even jobs like growing new limbs or ears or fingers. These nanocomputers will be able to do these specialized jobs because they can be programmed with the codes needed to work on this basic atomic molecular level, similar to the way nature has coded living structures for 4 billion years. While making change in a living organism's body cells, curing plants or animals of disease problems, for instance, is coming and is generally welcome on Earth. Making changes, however, in the germ cells, the sperm and egg, is more controversial. Changes here, whether done by gene guns or still to be developed nanotechnology, are permanent changes, passed on to offspring. Changes like nature has been making using mutated genes and natural selection pressures for 4 billion years. But now changes made by conscious, intelligent choice of one particular product of that natural evolution, human beings. If these changes accelerate in the third millennium, as they almost certainly will, they may result, as they have always done in the past in nature's evolution, in new species of plants, animals, and humans being produced on planet Earth. How these new varieties of human beings would cooperate or conflict with descendants of present-day Homo sapiens is unknown at the present time.